Yes, so as I mentioned, I'm Charlie. I'm from Dr. Rui Kwong's lab at the University of Minnesota. And today I'm going to be talking about our work in spatial transcriptomics, and specifically with this graph-guided Tucker decomposition, which we use for deciphering high-order structures in spatial transcriptomes. And I'll just give a very brief introduction to spatial transcriptomics. Spatial transcriptomics allows for the spatial resolution of gene expressions across tissues. And one of very popular technology is an in situ capturing method, 10x genomics Visium, which you can see on the right here. And so what this allows is us to map captured mRNA molecules within tissues back to spatial coordinates in the cells they were captured in. And so some of the reasons why these particular technologies are useful is something like tissue segmentation, where we can spatially visualize how cells and spots in the tissue are grouped together and share similar gene expressions. We can also do some sort of visualization and identification of marker genes such that we know where they're being expressed in the tissue. And we can also look at the spatial components, which are essentially looking at groups of similarly expressed cells or some sort of underlying structure of how genes are being expressed throughout our tissue. And so some of the concerns for these spatial transcriptomics data analysis and modeling them is taking into account this low capture efficiency, especially in situ capturing methods. And so this low capture efficiency leads to sparse gene expressions. And obviously this can cause some difficulty in certain analyses. One thing we also want to consider is the sort of natural spatial structure that needs to be captured in however we're representing our spatial transcriptomics data. And so one way we can actually account for this spatial structure is through the representation of our data as a tensor. So this is just a multi-dimensional array. And this allows us to preserve the high order structure that is inherent to this data. And so given these properties of the data, the ST data that we're looking at, it's important, I guess, where you can ask, how can we analyze these data sets when they're large, they're sparse, and but we also need to take into account their capturing inefficiency, the spatial structure, as well as the size of them. And so one way we can approach this is through factorization of the data. And so this can be done whether the data is represented as a matrix or a tensor. And so just for example, some of the ways we could factorize spatial transcriptomics data is through a non-negative matrix factorization, which is been commonly used before. There's also another option would be non-negative matrix trifactorization. But of course, if we have our data represented as a tensor, there's also tensor decompositions that we call them to decompose, factorize our data into low-level components. And so a very popular tensor decomposition method is this canonical polyadic decomposition, or the CPD. Another one is the Tucker decomposition. And so we're interested in this Tucker decomposition, which I'll go into specifics here, mainly because it's able to capture multilinear interactions among its components, which the CPD is not able to capture. And it's also not been applied to spatial transcriptomics data before. And so what we wanted to do was use a Tucker decomposition model to develop a model that can find low-level representation of the data that also captures biologically important spatial structures inherent to the ST data, while also preserving the natural high-order structure. So given this, I'll introduce our model, which is Graph Tucker, which is short for Graph Regularized Tucker Decomposition for Higher Order Analysis of Spatial Transcriptomes. And so just I'll go into more details about the actual optimization of this model, but I'll just provide a high overview. So this model consists of a Tucker decomposition. So this is a decomposition of our tensor, as well as this graph regularization term. And so the introduction of this graph regularization term is useful because it can help us enforce some important biological and spatial assumptions about the decomposition that we're looking for. And additionally, since spatial transcriptomics data is count data, we want to enforce this non-negativity constraint on the decomposition we're looking for. Okay, so of course, we need to understand what is the Tucker decomposition. So the Tucker decomposition is generalized to tensors of any dimension, but specifically with the data we're working with, this is three-dimensional data. We have an x-axis, a y-axis, and then an axis corresponding to the genes that we captured. So the Tucker decomposition decomposes a tensor into three-factor matrices, one corresponding to each of the dimensions. So we have AX, AY, and AG. And each of these factor matrices has some number of columns, which we call components, and they have their individual ranks. And most importantly, this Tucker decomposition also introduces this core tensor G, where the entries in this core tensor are going to store the level of interaction between all combinations of components between the three-factor matrices. And so one nice part about applying this decomposition to spatial transcriptomics data is that these components are inherently interpretable. So just for example, if we look at these AX and AY components, so that correspond to the two axes we're working with, they're capturing some sort of spatial information that's present in the data. And additionally, our matrix AG, which corresponds to our gene components, and these can be interpreted as gene modules. Another thing I want to mention is, sorry, it looks different. When the core tensor is superdiagonal, specifically when the superdiagonal of this core tensor is enforced to be non-zero along the superdiagonal, the Tucker decomposition and the CPD are actually equivalent. So what this is saying is that under certain conditions, the Tucker decomposition can sort of be interpreted as a more expressive version of the CPD. And so for actually computing this non-negative Tucker decomposition is relatively straightforward, and it just involves minimizing this squared loss term. And we also introduced this binary mass tensor M, which is mainly used to select training and testing entries in our original tensor that we want to include in our optimization. And this is mainly used for cross-validation experiments for testing imputation of missing entries. And moving on to the second portion of our optimization is this graph regularization term. So the main reason we want to introduce this term into our optimization is sort of twofold. We want spatially adjacent spots to share similar gene expressions. We also want functionally related genes to share similar expressions. So how we do this is we take the Cartesian product of three graphs, a spatial chain graph in the X dimension, a spatial chain graph in the Y dimension, as well as a protein-protein interaction network corresponding to our gene dimension. And so by taking the Laplacian of this Cartesian product graph, we can actually introduce it into our optimization problem such that we're enforcing those two assumptions on our decomposition. Additionally, we have this hyperparameter lambda, which allows us to tune the level of trust we want to put in our network and the effect it's going to have on our decomposition. So looking at the optimization as a whole, optimizing it is relatively straightforward, and we use an alternating multiplicative updating approach, which allows us to maintain non-negativity. And this basically works where we update one factor matrix or the core tensor at a time while fixing all the other components. And I won't show them here, but you can explicitly write out the update rules for each of the factor matrices and the components. And one important part of this model that may not be apparent is actually the interpretation of what we call the spatial components. So given that we've run Graph Tucker on a spatial transcriptomics data set, we'll obtain this graph regularized decomposition of the original data set. And actually, if we combine the core tensor with our two-factor matrices corresponding to the AX and AY dimensions. We can construct this sort of subtensor, GXY, where each frontal slice of this tensor corresponds to what we call the spatial components. So given we construct this, we can visualize where each of our gene modules are being expressed throughout the tissue. And this is just an example of the retrieval of this spatial component that's corresponding to this region of the heart in this mouse embryo. Okay, so given our model has the ability to extract these important spatial components that might represent spatial domains in the original data, we wanted to evaluate and quantify how well these spatial components are capturing known regions of tissues. And so we did this through two main experiments. So first is this spatial domain recovery, looking at the individual spatial components. And so we did this by measuring the AUC and the Euclidean distance between each spatial component, as well as a ground truth region and pairwise, and then selecting the match that had the lowest Euclidean distance. And the idea behind this is that we want to find the spatial component that does some sort of best thresholding and is a best representation of the region that we're looking for. And in the second experiment, we wanted to observe how the spatial components are capturing the regions as a whole, which we did by performing clustering on the spatial components across their spots. And so the idea behind this is that it should indicate whether our low-level representation of the spatial regions are still capturing important information for delineating these tissue regions. So just to quickly go over the data sets that we used for our experiments, we used this human breast cancer and human brain data set for, that have ground truth annotations of the regions for our first experiment, and as well as the second experiment. We also did another experiment just to evaluate the scalability on high resolution data set on this mouse embryo stereoseq data set of embryos from 2 different days in development from the MASTA project. We also did a imputation experiment, but I won't talk about them here. And just to quickly go over the baseline comparison methods, we compared to just using something like non-negative matrix factorization, regularized non-negative matrix trifactorization, very recently developed non-negative spatial factorization that allows for similar extraction of spatial components. And we also compared it to a tensor-based method, which is FIST. And so here I'm providing an overview of the quantitative results. And I just want to point out the most important part, which is that when we run Graph Tucker with this graph regularization included, our spatial components are, on average, having higher AUC and lower ED compared to our other methods, as well as higher ARI in terms of correlating to the original clusterings of these data sets. And another important thing is you'll notice that we have this Graph Tucker n%. So one benefit of using the Tucker decomposition is that we can sort of choose the top threshold of highest entries in our core tensor. So choose the top % of interactions to include in our evaluation. And so we actually found that when we include the most important interactions, we're actually getting better spatial component matches to the regions. So here are some visualizations of our results on this breast cancer data set. And so you can see just on a visual comparison, regardless of the ARI, that Graph Tucker is doing a good job of recovering smooth clusters that relatively correlate to the original ones, especially compared to the matrix-based methods. And also want to point out some good examples of 8 of the 20 regions here of the spatial components. So specifically this DCIS, LCIS 1, 3, 4, and IDC 5. You can see that the spatial components at the bottom are doing a good job of uniquely capturing their corresponding regions, as well as finding smooth representations of these matches as well. And so additionally, on this other human brain data set, I just want to show again, the ARI is higher. Most of the spatial components, as you can see, struggle to recover these clusterings. Though you can see that Graph-Tucker is doing a relatively better job at capturing this L6 region in its clusterings. As well, I want to point out some of the best examples of Graph-Tucker, which are these L3 and L6 region that are relatively accurately capturing these regions. And so lastly are these results on this high-resolution stereoseq mouse embryo data set. And for these data sets, we don't have the spot annotations such that we can't do the spatial component quantification to determine matching. So these were based off visualization alone. And so you can see, I just want to highlight the regions that are present across both of the embryos. So you can see this brain area, the branchial arc, the dermomyotome, the heart, the liver, and the sclerotome are both accurately being captured by some of the spatial components from Graph-Tucker. And so some of the major takeaways. So Graph-Tucker provides a high-order factorization model that allows us to capture multi-way interaction among its spatial and gene components. And we saw that this graph regularization term is important in finding not only biologically meaningful spatial components, but also such that we can accurately delineate these spatial domains. And then lastly, in the experiment we just saw, Graph-Tucker is scalable for larger scale, high-resolution ST data such as stereoseq, which will certainly be more important in the future. And so I just want to thank my advisor, Dr. Rui Kwong, as well as my lab mate Tianxi Song. This definitely wouldn't happen without their help and guidance, so I appreciate their help. And thank you all for listening.
Thank you, Charles. So we have time for questions.
Hi, great work. So one question is that, did you see that the things that you were detecting is a mixture of cell type, or it's like what's its correspondence with the cell types that are annotated? So for example, in the embryo, the domains are not identified, but I guess the cell types are identified.
Yeah, that's a good question. Actually, one thing that we saw was that, especially in this breast cancer data set, was that certain spatial components may correspond to more than one region in the annotation, which sort of suggests that perhaps some of these regions or these cell types are not completely separable based on maybe their spatial properties alone. And the signal to distinguish something like cell types, it wasn't something we were looking into directly, but it would be something to look for in the future.
Hi, thank you for the wonderful talk. I was wondering whether the graph regularization actually imparts something biologically meaningful during the factorization, or it could have been just a regularization that could have happened in a random graph. And then if it was like biologically meaningful, I was wondering if there is an interesting insight that came from that specific component of your factorization method.
Yeah, so I think one thing in general is that from all of our results across all the experiments, we saw that the graph regularization was helping both in the imputation of in our cross-validation experiments, so that would involve removing specific spots. And in terms of the biological importance, I didn't show them here, but we did evaluate some of the marker genes for these corresponding spatial components and some of the matched regions in the human brain tissue. And we saw that these marker genes, when ran with the graph regularization, were being highly ranked, as we should expect.
Hi, thanks. Great talk. So could you say something about the computational complexity of just not the Tucker decomposition, but also the optimization problem that you're trying to solve with the graph regularization, and when you compare that to the baseline, and what's the dimension of the data, something along those lines?
Yeah, so I guess the first thing is that one of the hard parts when dealing with tensor decompositions is that choosing the rank for your factor matrices can be hard. Obviously, that's not something we were trying to solve, not that it is solvable. But that sort of adds some additional complexity into knowing what rank you need to choose given your data, and especially for this large data sets, choosing too high of a rank can significantly increase runtime because you are storing so much more components in your data. I guess in terms of the size of the data, I would say compared to the other methods that we were comparing against, there was no like large significant need for I guess like extra storage or concerns with the runtime. That was only really present in the larger of the two StereoSeq datasets that is significantly higher resolution. I think it's like 150 by 150. But even then, it was still able to run where some of the other methods that we compared against were not scalable to this size.